I have no conflicts of interest. I'm Jonathan Richard from Andres Finzi Lab, and today I'm really happy to present our latest work in collaboration with Daniel Kaufman, demonstrating that productively infected cells are resistant to non-neutralizing antibody. So we know that the HIV-1 envelope conformation substantially impact antibody recognition and ADCC responses. So in its unligand form, the envelope adopts a close conformation that can be preferentially targeted by broadly neutralizing antibody, but not non-neutralizing antibody. These non-neutralizing antibody target epitopes that become only exposed when the envelope adopts a more open CD4 bound conformation. These non-neutralizing antibody target also cells that are coated with GP120, so the exterior subunit of the HIV-1 envelope. This is not the case for the broadly neutralizing antibody that either compete with CD4 for the binding of envelope or target epitopes that are only present in the trimeric envelope. ADCC-mediated non-neutralizing antibody are naturally present in the sera from infected individuals. Consequently, the virus evolved several strategies to prevent CD4 envelope interaction. Notably, the virus downregulates cell surface CD4, which is done by the VPU and Env protein. This forced the envelope to adopt its close conformation, thus protecting HIV-1 infected cells from ADCC mediated by non-neutralizing antibody. However, GP120 shedded from an infected cell can interact with CD4 on the uninfected bystander cell, so presenting CD4-induced epitope and sensitizing these uninfected bystander cells to ADCC responses mediated by these non-neutralizing antibody. So despite significant advances on the understanding of HIV-1 resistant to ADCC, the capacity of non-neutralizing antibody to mediate ADCC against productively infected cells remain controversial. On one hand, several studies demonstrated that wild-type infected cells are protected against ADCC responses by these non-neutralizing antibody because of the action of Env and VPU. But on the other hand, studies reported that non-neutralizing antibodies such as the A32 antibody could mediate ADCC responses against HIV-1 infected cells, suggesting that CD4-induced epitope could be exposed on infected cells, notably on a cell population characterized as P24 low CD4 positive cell. In this study, we decided to use flow cytometry and RNA flow FISH techniques to characterize cell population targeted by broadly and non-neutralizing antibody in a context of infected primary CD4 T cell. Activated primary CD4 T cells were infected with the transmitter founder virus CHAV-77, and 48 hours post-infection, cells were stained with a panel of broadly neutralizing antibody versus non-neutralizing antibody. Cells were separated based on cell surface CD4 and intracellular P24. You can see that cells that are P24 negative and CD4 positive were efficiently recognized by the non-neutralizing antibody here in black, but not by the broadly neutralizing antibody here in green, confirming that these uninfected bystander cells are coated with shed GP120. Cells that were found to be P24 low and CD4 positive were also more significantly recognized by the non-neutralizing antibody, but the weak recognition by broadly neutralizing antibody suggested a mixture of shed GP120 and CD4-bound trimeric envelope. Cells that express high level of P24 and low level of CD4 were found to be more significantly recognized by the broadly neutralizing antibody over the non-neutralizing antibody, suggesting that these cells present envelope in its close conformation. We were able to detect a strong recognition of P24I with non-neutralizing antibody only when we use a virus that is defective for Env, so unable to fully downregulate CD4, so exposing envelope in its CD4-bound conformation. And in that context, we detected similar level of detection by non-neutralizing antibody and broadly neutralizing antibody. We also look at Env expression using a Env defective virus as negative control, and you can see that we detected Env expression only on cells that are P24I CD4 negative, so the cells that are targeted by the broadly neutralizing antibody. So this suggested that broadly neutralizing antibody targets cells that are CD4 negative, P24I, and NF positive, while the non-neutralizing antibody targets cells that are CD4 positive, either P24 negative or P24 low, and NF negative. So to identify which cell population is productively infected, we next use the RNA flow-fish technique developed by Daniel Kaufman in Montreal to determine which cells are enriched with envelope and NF mRNA. Using this technique, again, the cells were characterized based on CD4 and P24 expression. You can see here that the majority of the cells that were CD4 positive, either P24 negative and P24 low, were negative for envelope and NF mRNA. In contrast, the majority of the cells that were CD4 negative, either P24 low or P24I, were positive for envelope and NF mRNA, while a maximum of double positive cells were detected among the P24I cell, confirming that CD4 downregulation precedes the expression of HIV-1 late transcript, and that productively infected cells are CD4 negative, so less likely to expose CD4-induced envelope epitope. Then we wanted to evaluate the capacity of a non-neutralizing antibody A32 and a broadly neutralizing antibody PGT126 to recognize the cells that are either negative or positive for envelope and NF mRNA. And as you can see here, the majority of the cells that are negative for envelope and NF mRNA were efficiently recognized by the non-neutralizing antibody A32, but not the broadly neutralizing antibody PGT126. Inversely, cells that are productively infected, so positive for envelope and NF mRNA, were efficiently recognized by the broadly neutralizing antibody PGT126, but mainly resistant to recognition by the non-neutralizing antibody A32. We also specifically look at the level of envelope mRNA among the different population. And while the cells that are targeted by the broadly neutralizing antibody PGT126, so the cells that are P24I and CD4 negative, express high level of envelope mRNA, cells that are targeted by the non-neutralizing antibody A32 were negative for envelope mRNA, suggesting that these cells do not express HIV-1 envelope and most likely represent cells that are coated with SHED-GP120 or non-infectious viral particle. To determine the capacity of broadly versus non-neutralizing antibody to target productively infected cells in vivo, we isolated CD4 T cells from 6 chronically infected individuals, and upon activation were able to reactivate the endogenous virus as detected by P24 staining. Cells were then characterized based on CD4 and P24 expression, and they were stained by a panel of broadly versus non-neutralizing antibody. As we observed for in vitro infection, cells that were CD4 positive, so either P24 negative or P24 low, were found to be more significantly recognized by the non-neutralizing antibody over the broadly neutralizing antibody. Inversely, cells that are known to be productively infected, so CD4 negative and P24I, were found to be more significantly recognized by the broadly neutralizing antibody. Consequently, these cells were more susceptible to ADCC responses mediated by these broadly neutralizing antibody and mainly resistant to those mediated by the non-neutralizing antibody. In conclusion, we demonstrated that productively infected cells are preferentially recognized by the broadly neutralizing antibody. These cells efficiently downregulate CD4, express high level of NF and P24 protein, and are enriched in HIV-1 mRNA. In contrast, cells targeted by the non-neutralizing antibody or CD4 positive express little or no P24 and are negative for NF and HIV-1 mRNA. These cells are envelope mRNA negative, suggesting that they are coated with SHED-GP120 and or non-infectious viral particle. We show that CD4 downregulation precedes the expression of HIV-1 late transcript, suggesting that the productively infected cells are CD4 negative, so less likely to expose CD4 envelope epitope. And we demonstrated that ex vivo expanded CD4 T cell isolated from infected individuals are sensitive to ADCC mediated by the broadly neutralizing antibody, but resistant to those mediated by the non-neutralizing antibody. We believe that this information is important for the development of immunotherapy-based strategy aimed at targeting and eliminating productively infected cells. Finally, I would like to thank everybody that contributed to that work. My director, André Spinzi, our collaborator, Daniel Kaufman, Jérémy Sagnier, and Mathieu Dubé, who generated all the RNA flow fish data that I presented today, our founder, and you for your attention.